This way I can send the link. Okay, share screen, portion of the screen, share. Okay, all right. Now you'll see the green border. That is the one that I'm trying to share, okay? Okay, I will center that in just a second. Okay, it flipped back. Okay, this is where we had left up. We had done about 75 slides. So we talked about briefly about the two big species, the Neanderthal and Denisovians, that were discovered in a small cave in Siberia from just a little pinky of a bone, just to refresh your memory type of thing. Now, again, just to give you a refresher, this is the classic Darwinian evolution that all of you learned in school a long time ago. It's a heritable variation, which is spontaneous and a relatively slow process that requires long duration of time. And this way one can, any being adapts to environment, the classic Darwinian beaks. And this was the famous statement, just to refresh, from beginning of origin of life, from so simple a beginning. That's the classic statement of Darwin. Okay, and just to refresh our memory, this is the two big forces, natural selection and genetic drift, which is pure random. So the way this is genetic drift is, as I may have explained, is probably just random and stupid. It just happens by chance. Okay, so and natural selection is very deliberate and ruthless. Those are the two words that one evolutionary biologist I heard in the talk, and which makes sense, in the sense that it will eliminate the bad mutation that has taken place and keep the slightest advantage. And as I may have emphasized, nature uses what she has, does not reinvent the wheel, so to speak. So those are the basic principles. Now, it is interesting, the power of computing. Okay, this is because I was researching genetics. Google News pushes these items to me. So this came across just 2 days ago. This is an article that was published in the Atlantic magazine, how a rare brain mutation spread across America. And it's almost like a scary thing as to how this is called in genetics. The geneticist or the molecular biologist use a word as something called the founder effect. It means a one band mutation if they have a lot of children. And what happens is that the mutation does not manifest until you've had children. Okay, so some of you were in my aging class and remember that is called anisotropic pleiotrophy. Means the good mutation, it stays because you have the children. It doesn't affect having the ability to have children, and it shows up afterwards. So this is a mutation that took place from one couple that was in this 18th century in North Carolina. And they had a rare mutation that caused weakness of the blood vessels in their brain, so they could bleed. And this particular mutation has been traced in all across southern and western United States. And when by chance they found it in one family in North Carolina, and other people appeared, and that the data was shared and said, okay, well, they are not related, but somewhere along the line, they must have been related to each other. So this is how this rare mutation was passed on. And I have some other examples of this as to how mutation can survive, even though, and just by having a lot of kids, so to speak. So, okay, now we talked a bit about Neanderthal, and I mentioned that if you have your blood test done through 23andMe, you will find out if you are, other than pure Africans, you will have some Neanderthal genome. And I had done that earlier, and I have bought, like, as usual, like 2.5 to 3% or something. Okay, but there was a lot of interbreeding that we talked about, about amongst all the species, so there was a lot of hanky-panky going on in the ancient days, so this is, and so there are a lot of base changes, single base changes, as you may recall from last conversation, that DNA has 6 billion letters, and how many single base mutations. A lot of them are sort of meaningless, but there are as many as 96 amino acids that are different. And it takes various length of amino acids to create a protein, which is the workhorse of human tissue. So there is effect of Neanderthals that is still lingering on in our genome after so many thousands of years. Now, some of the mutations that we may talk about may surprise you: skin color, hair, chronotype. By chronotype, I mean, I did a class on sleep, means, like whether you're an early riser or a night owl, so to speak. So that, and some of the serious diseases such as lupus and biliary cirrhosis, at least portion of it may be related to some genes that you inherited from Neanderthals. Okay, at any point, if anybody has questions, please raise your hand. Now, I'm cheating because I have my notes on one side, and so I can't see everybody's faces that easily. But so, okay, I think if anybody has questions, just interrupt and say, because I will try to keep the participant window open, but I can only see one this thing, so I have to scroll back and forth. So, and if anybody else wants to be a co-host, I will be glad to make them a co-host, so they can be so-called gatekeeper.
What is DM?
DM is diabetes mellitus. I didn't want to type the whole thing, so I just typed diabetes mellitus. So at least some tendency toward diabetes may have been inherited. That obviously it is what we call a multifactorial, or to use a fancy word called polygenic disease. So at least some element came in from because of living in the cold climates, And there is an interesting book that I read it as an ebook, which is written by an evolutionary physician. It's a Israeli name. I can't pronounce the name, but it is called The Survival of the Sickest. It's an interesting book, I think that she talks about how living in the cold climate might have triggered diabetes, because, and this is to be taken a little bit, with as a hypothesis rather than through this thing, as to how living in cold climates, and sugar becomes like an antifreeze, so to speak. And actually, there is actually a small frog that literally, when the weather gets cold, it freezes its whole body. It almost becomes like a rock. And when it warms up, it has like a special antifreeze protein. So, but that we are digressing. We are not talking about other species. So we'll concentrate on humans and genetics. So these are some of the genes that, and I talked about the physical features last time, so I'm not going to repeat just for sake of time. And so, and there are some of other factors, that are also may have been, like some of the autoimmune diseases, and lupus is obviously one of the big autoimmune disease, where your body loses the identity of self versus non-self. So that's where it comes into picture. So there are a variety of diseases which might, we can say that the gift of Neanderthals, so to speak. And we talked briefly, you may recall, the Denisovian gene had helped the Tibetan population. Okay, and now this was an interesting slide as to how did the Neanderthals and Denisovians smell, So this was different sense of perception. So they were more sensitive to smells associated with honey. And this is how they figured this out. Don't ask me, but apparently this was, there is some genetics. This shows the genetics was interesting. How, I don't think there's a living Neanderthal, so how they can say sensitive to sulfurous smells. That's I didn't quite understand that, but it may have to do with the various hormones. And so this is, at least, this was one of the findings.
And Barack, Cliff Davis, testing. Can you hear me?
Yeah, please go ahead.
Yeah, I had a question about the lack of Neanderthal transitioning from what was before to current, and the fact that African Americans don't seem to have there's a small percentage, I guess, that has that. What's the transition from before to current Homo sapiens for those individuals that don't have the Neanderthal connection?
It was a totally separate tree, not unrelated. They're like, it's like when we talked about, it was not a one single tree, which was like a logical ladder-like transmissions. There are several, I have some other slides about the other species that just evolved spontaneously. So there are multiple branches that took place, Now some did not survive because of climate change and other things and just petered out, It's just, now this, we just talked about three separate things. So it just occurred spontaneously because Africa is a wide continent. I mean, you can put the whole of North America, Canada, and Brazil inside Africa. So spontaneous evolutions took place to Homo sapiens. That's why if you recall, there was a slide about there's a lot more diversity within African tribes itself. So that's because they've been around for a lot longer. So it's like, for example, you take somebody from Nigeria to somebody from Zimbabwe. There's a lot more diversity within the groups, type of thing. So there are two totally separate evolutionary trees. It's like they are not all classic like a pine tree that Homo sapiens are at the top. They're like a varieties of. I want to say, let me see if I can give you an analogy. It's like more like a big, huge banyan tree, which is with different branches all over that occurred, type of thing. So that's it. So, I mean, I can't come up with the exact analogy from the plant world, but, so there are separate evolutions that took place.
Thank you.
So I mean, so it's just like, so we, because that was the classical view that was upended that, because we'll talk a little bit about the hobbit or the Homo floresiensis. So the slide is just coming up, because I did not get to it last time. I was hoping to cover it, but now this was a fun slide. This was an article from the British magazine Guardian. Okay, the new research shows their distinctive features. You can see how the nose is, and a lot of Asian Indians, at least some of the Asian Indians have huge noses. Okay, and now I think that India is becoming a little bit more rich. There is a trend for plastic surgery also, type of thing. And some people, so this is this huge nose type thing. So obviously, this was a commercial from some bank, that I saw it in one magazine. So I said, well, you look at the nose. I mean, you can say like, if you put this guy in the modern time, you might be at home. So it's just an interesting thing as to how, and I've seen some Indian movie actors, their noses, I mean, like, and even some of the Iranian noses can also be big type of thing, or, Caucasian type. So that's varieties of noses, but, that gets into as to some of the features might have been, and some of the stuff also like prognostic, like a prominent jaw. So, and I'll talk a little bit about how's the genetic genes affect the development of the various body parts. So we'll get to that in just a few slides down the line. So these are the varieties of explanations that have. Now, this is an interesting that the research is fascinating. That just on July 3, I just came up with an article while I was reading up vigorously on the SARS-CoV-2, and that a risk factors in the SARS-CoV-2 was inherited from archaic Neanderthal genes. And it occurs at a frequency of 30% in South Asians. Okay, so this was, and because the Brits reported that Britain has a large population from former East Pakistan, which is now Bangladesh, and they had a much higher mortality in that particular ethnic group. So based on that, they were able to see that there is some tendency, a risk factor that was much more prevalent in the South Asian populations of Bangladeshi origin. So that made them at a much higher risk for getting a much more serious illness from SARS-CoV-2. And amongst the European population, it was only 8%. So So it just, so our ancestry still comes back to help us and haunt us, so to speak. So, okay. And this is, I did a class on single gene mutations type of thing, and the red hair mutation, that again, some of the, I think, even Neanderthals, the article from the Guardian magazine showed that some of them, they may have a darker skin with red hair, so that the mutation may have appeared, earlier and several times, And there is something called the MC1R. So the name is called melanocortin, and melanin is the pigment in the skin. So if a mutation takes place, then you don't form the pigment, so to speak. So, and they are very sensitive to light, as you very well know, more sensitive to analgesics. So these are some of the data that is, this gene is located on the surface of the melanocytes, which are abundant in your skin. Okay, so this is, so that was an interesting diversion slide type of thing. Okay, and there are varieties of other this thing being that there are some interesting genes that when you talked about, these are just of appearance. It doesn't have any real bearing on, health and or longevity type of thing. So there are, so one can find out as those varieties of genes still have effect on this. Now, just for completeness of sake, there are other species of humans have been discovered. And this was a fascinating story that American paleontologist who did most of his work in South America, Southern Africa, sorry, his son, who was like a 9-year-old, he found this fossil. This was actually genuinely a clavicle. So, Dad, I found a fossil. And so this is, and then they did excavations at that site called the Cradle of Human Civilization in South Africa, I think, and they discovered the entire species. And that is this this is a fairly recent discovery, I think 2009 or 2013. I forget, I don't have the exact date written down in my notes, but sorry about that. But so this is so this has been around for, and so this is a fascinating story as to how this discovery came about. And then they have discovered a huge number of fossils at that site, not just and almost like an entire skeleton, and that species has been named Homo naledi. Okay, and again, so this is, where do they fit in? These are just these examples of varieties of other species, the Homo habilis, so-called handyman, Homo erectus in between. And you can see, just for completeness sake, there are varieties of, so the brain volume was relatively small, okay, compared to, so, and smaller teeth. And you can see that the transition towards modern humans is taking place, and the flex occiput means, so it's that the spinal cord is coming down lower type of thing. So this is, so that means they were able to walk on 2 feet. Okay, so, and so this lived about 300,000 years ago in South Africa, a very projecting face, slender body with wide hips, human-like feet and hands. The brain was rather small, only about 500 cc, and weighing only about 39 to 55 kilograms, with a combined diet. And the species was named, this is, I have it in my notes on this slide, 2015. And Naledi means star in South Africa. It's one of the Sotho language from South Africa. And so this was discovered in the Rising Star Cave system where it was found. So this was a very groundbreaking discovery. If you leaf through your Smithsonian or National Geographic, you may have come across that. And this is, for sake of time, I wasn't going to shoot too many videos, but this is worthwhile. Okay, this is the type of the cave that they had to go through. This was, hopefully you can see how they had to crawl through this this tiny area where it's just barely shoulder width area. And so much so that the average male could not get through that.
6 remarkable young scientists squeezed through a 12-meter crawl down a chute 18 centimeters wide to get these fossils of a new species of early human ancestors, Homo naledi.
It's really unusual to see all women scientists in these kinds of situations where you are expected to enter into and work within what might be considered a fairly risky or dangerous situation. Ordinarily, it's the men jumping at these things, but I think because of the size limitations on getting down into the site, women were given more of a chance to sort of get their foot in the door.
Hello, Command Center. This is Maureen at the top of the chute. I'm just about to descend. Okay. Thank you. Bye.
You start by descending down a fairly narrow shaft and some tunnels. You have to crawl on your stomach for about 3 meters. Then you enter into another chamber. This is what we call the Dragon's Back, with the 4 or 5 meter drop on either side. At the top of the chute, you start the 12-meter descent into the chamber. You then go through another passageway into the main fossil chamber. The first thing that came through my mind when I went through the final slot was Howard Carter opening Tutankhamun's tomb and Lord Carnarvon saying, "What do you see?" And Carter says, "Things, wonderful things."
Wow.
God, this place is beautiful.
There is no find like this anywhere else. This is extraordinary on every level.
It's almost hard to put into words what this is going to mean for the story that we tell ourselves about where we came from. We're really after this story. This is what excites us.
It's not entirely clear at this point how it got there. They're so unusual. It doesn't seem to fit any currently known paradigm for fossil hominins. Unfortunately, the level of CO2 within this particular chamber of the cave system has spiked to a critical point, so we need to leave so that we're not all suffocating.
We need to get to the surface. CO2's up to 1,300.
Those first couple of days were probably some of the hardest, most difficult days of their life physically, because I was scared to leave people down there for too long. I was trying to rotate them out, which forced them to climb in and out this torturous path.
Okay, so you get the idea. So
6 remarkable young scientists.
okay, so it's an interesting this thing as to how this lady's found this thing. So it was his son's discovery was enough that 60 Minutes did a whole piece on it about this. They interviewed the son type there. So this was, so one can find it, I haven't put in a link for length of time. So these are the similarities. So it was a remarkable discovery. Okay, and again, not to be outdone, this was almost broke, because obviously the earlier paleontologists thought that the settling of Americas was fairly recent. But just recently, somebody has discovered footprints in New Mexico that dates back the time to by several thousand years. So new colonization has taken place. So the varieties of, and they may have traveled not either earlier by the land bridge or across the Pacific type of thing. So it still wasn't settled, but this was almost a mind-blowing event. And we already talked about how the Australias were settled a lot earlier because, and I'll have some more slides on that afterwards. Okay, so we just walk basically, okay, or by a canoe. It hasn't been settled actually how it came about. And this was the other discovery that we have talked about in 2003. And usually in the wet, humid climates, it was very hard to discover fossils because everything decomposes very quickly. And it's almost impossible to get DNA from that, in contrast to Siberia. So this was a species that was discovered in a tiny island in the Indonesian archipelago in 2003. And that has been dated anywhere from, so almost like 100,000 years ago. I would say a tiny person. It was obviously, with the popular stories, it was named Hobbit promptly. And again, and there were a lot of other wrong ideas that were floated earlier, that these people were deficient in thyroid, or they were not really even human, so that type of thing. So, but the more recent discovery have showed that that they did not have any abnormalities of their iodine metabolism or nutrition or anything else. But so they were, and They were fairly sophisticated, this thing that they had stone tools. They hunted small elephants, which was stegodon. And now it's interesting that when animals evolve in a small resource area, small animals tend to be big and the big animals tend to be small. So the elephants were small, but some of the other animals were fairly big. So this was, and some of the remnants from that era might still be the Komodo dragons, okay, from that area, but they have not survived, and because of the climate difficulties, they haven't been able to isolate the DNA. But this was known as Homo floresiensis, aka this was the nickname was given Hobbit, like Lucy was given earlier. Okay, and just to give you an idea, this is Australia, Borneo Island, and if I can get my magnifying lens, this is where Flores is, So this is Sulawesi, okay, and East Timor, okay, and so this is between Borneo and East Timor. So this is the island of Flores, and these are some of the caves where these discoveries were made. Okay, all right. So, and this is the so-called Wallace Line, because in the earlier days, people were able to walk because of the land bridge. Okay, and this is, so we get around the globe, and the Australia was settled much earlier, and these were some of the fossils that were discovered, and the date has been pushed back further and further, is to 65,000 years. So, and again, they're not to be outdone. There are some other species that have been discovered in the Malaysian area, okay, and some of the caves in Borneo, as well as in Philippines. So varieties of separate evolutions of humans have taken place. That's the point that one wants to get across. Okay, and this was again, in Ethiopia, new skull was discovered, and so that dates back to 600,000 years ago. So again, so you get the idea that there are a lot of separate evolutions, and the Chinese have been doing a lot of excavations in their area. So there, as recently as this year, so some of the older skulls have been discovered, in the maybe mispronouncing it, Hebei province, and they named it Homo longi, which means dragon. So the dragon man, But some of the experts are contesting that these may turn out to be Denisovians after all. So that is still a point that hasn't been well settled, and so this may be so maybe a relative of humans rather than to Neanderthals. So, but the Chinese paleontologists, they do some really impressive work, and for those of you who are more interested, these are some of the links. All these links that are pushed in, they are active links, and if you're curious, you'll be able to read the original articles based on that. Okay, please interrupt me as you have any thoughts come up or questions or comment. Okay, and some of you may recall we talked about the Darwinian evolution, and with the rise of genetics, that was called neo-Darwinism. But things have gotten in biology, things are a lot more complicated than in physics or mathematics, for example. And now we have learned a lot more that it is not a nice, smooth, long-term evolution that takes place. But things are a lot more complicated, that the environment has a lot of influence on human evolution. I may have given you some examples, starting from the birds that are run over by automobiles, or the elephant tusk that is being reduced because of the poachers. And the same way evolution takes place in humans also. And the classic example was during the hunger winter in Holland that took place, when during the Nazi occupation of Netherlands, when the food was scarce, and then the next generation became more obese because of starvation during pregnancy, and because of survival and interestingly, this effect continued even in the third generation. And I'll have a lot more to say about the new science of epigenetics. And this is kind of the revenge of Lamarck. If you may recall, Lamarck was poo-pooed in our science textbooks, at least in mine, that Lamarck was all wet and Darwin was the king. But it turns out that some of the so-called epigenetic effects. Epi means above, genetics means genetics. Means the DNA code actually does not change, but a very subtle biochemical changes take place in individual genes. So the expression of the gene is different. Means the gene is there, but it is either silent or it is expressed. And I'll have a few more interesting slides about how it goes on right before even you're born, that the X chromosome is silenced during fertilization, and so there are a lot of other factors. And now with the advance of computers, it becomes like basically it's, we're dealing with an integration system. And I think if we talk a lot more about microbiomes, in one class that we talked about, history of infectious disease, that we are not just isolated human beings, but we have millions and millions of bacteria in our gut, and yes, and parasites. If you have a cat, you're positive for a parasite called toxoplasmosis. Okay, so 90% of cat owners are positive for toxo. Okay, it doesn't do anything, unless your immunity goes down, then you're in big trouble. That's why a lot of people with HIV got not just rare uncommon pneumonias, but also got sign of toxoplasmosis. So the same way, there are parasites in your body and billions and millions of viruses. We'll have a lot more to say about viruses. There are some almost spine-chilling, goosebump-raising discoveries that are being made. So this is, and the concept is that we are all, instead of being a reductionist philosophy that we had, that trying to find an answer in a single 20,000 genes, they were so much interconnected with other species that is, and influenced by the environment, by the toxins that we may be influenced, and by our lifestyle. So this is an integrated, theory of evolution beyond Darwinism. So this, you can see how these circles are overlapped. Darwinism, variation, inheritance, natural selection is here. Gene mutation is somewhere here, modern synthesis. And the integrated synthesis, it incorporates plasticity, epigenetic inheritance we talked about. And so varieties of factors are overlapped circles. So these are some of the more modern thoughts about biology that takes place in. And this is actually the whole science. It is labeled as systems view of life. It's a whole integrated complex system. Okay, all right. And some of you who are probably more into math, may realize there's a whole science of complexity that has arisen since the late 80s. So this applies equally well in biology as well. The differences are, so these are some of the, as we talked about the systems view of life, so to speak. Okay. Okay, now again, so the body has two big plans, so now how does the body form? Our head in one place and feet at one side, and the two divided thing is to heart on the left side. So the body has a plan, which is called the Hox genes, and they have a small base pair sequence that's called the homeobox. Okay, and that creates various proteins, and that are folded in a different, rather complicated helical form that kind of winds around on itself and loops. And you can see how that, so that way a gene that is here can influence something that is here. It may be, if you draw me in a straight line, it may be a long distance away, several nanometers away. But when it is folded on itself, a gene that is here can influence the activity of the other ones. So it's a complicated scenario, or a dance, as you can call it. And this is conserved. You will be shocked to know that you're not, the body plan is not too much different than that of a lowly fruit fly. Okay, and so the normal fruit flies may have an antenna at the top. But if the scientist can manipulate the genes in such a way that can silence the gene or insert it in a different place, a leg can grow in place of an antenna. You can see how it almost looks like a bizarre or a monster. And one can see that the early embryologists, when they would see, and this may have something to do with the so-called cyclops, and the various, those of you who are in medical school, remember that in the anatomy museum, all sorts of fetal malformations were, saved, that's almost enough to give you nightmares, so to speak. So this is, now again, we'll see if I can zoom in without messing up myself. So these are the various Entonopedia cluster and the mid-thorax cluster. See how these are the segments that are devised, And so there are varieties of Hox genes that take place, Okay, now this nature has done varieties of experiments, and this was almost blew my chair. So what one would be surprised as to how much one can learn from reading The Economist, is to see how various animals, again, we're not talking about the whole animal kingdom here, but praying mantis, 1 ear, 2 wings, 3 hearts of octopus, 4 stomachs in cow, 5 arms. That is the radial evaluation instead of bilaterally symmetrical. Some of the spider eyes, huge. And even in our own body, it's hard to explain how the kidneys are paired organs, so are lungs. Spleen is there, but not a must. One can live without it. And there are organs that are relatively simple, such as bladder, which has only one type of cell line, basically, and the muscle at the end. It's like basically a storage bag. And at the same time, there is a complex organ, such as brain, which has several different types of cells and a complex neurons. So it depends upon the form has followed the function, so to speak. So that's the evolutionary beauty. Okay. Now, again, certain things are homologous, as to see, one can see that how cats are mammals, so are whales. Okay, so that is similarity. One can see that in whales, so that the humerus has shrunk, but it's there. So has the radius. Okay, and similarly, bats are mammals, so that's there. So this is the homology that it shows the common ancestry. One can say there's not much similarities between a bat and a human, but when you look at just the gross anatomy, not to mention the sequences, we're not even getting into the DNA sequences, as we showed you last time, the sequences, and I'll have a slide coming up on there shortly, I think so. So just goes to, this is the homology that is preserved just based on gross anatomy. Okay. On the other hand, there is analogous evaluation. For example, the wings in a fruit in a butterfly is a different evolutionary path than that of a bat. Okay, so these are the varieties of evolution from jawless fish that we talked about, which just has only a notochord, a simple, a nerve, but not vertebrae. Then the fish, and okay, now you can see how the evolution human Hox gene. So, and a fruit fly has only one set of Hox gene, but humans have several copies of it, and you can see how the varieties of copies are laid out based on color coded, And these are a series of slides that I cut and pasted just to show how it is preserved and across the whole animal kingdom. And mammals, you can see mammals and humans, not too much different. There are some differences, but one can just eyeballing, one does not have to be a geneticist to show that, that's quite similar. Okay. Okay, when you get down to chicken, you can see the differences are much more apparent. Okay, when you get down to the fish, you can see that the body plan is quite different in the so-called genes. So some genes are duplicated, some are eliminated, as to see how one can see that the genes that are essential are preserved, while the others are modified as the evolution proceeded. Okay, so that's the gene duplication. That is one of the big principles how evolution has taken place from the smallest organism to human beings. The other important point that some of you may be familiar with this remarkable scientist, but just she does not get enough credit, but she was, I may have mentioned it earlier, and obviously we talked about it in the history of medicine class, okay, that Barbara McClintock, okay, that working with corn or maize with of different color, she showed that there are certain elements that are actually transfer or jump from one place to the other. And she did her work in the early 30s and 50s. And her idea of jumping genes, as you can see that from this little cartoon on the side, hopefully I can zoom in on that. DNA called transposon, that's the same element as jumping genes in plain English, so it jumps from one place to other. So as to how disrupted gene, it kind of just jumps and inserts itself in another location. And this happens rather quickly. So how it is cut and these things. And that's another mode of evolution that nature has used shamelessly. And so it took about 30 or 40 years for her work to be accepted in mainstream. And because her work was not initially well received, she continued to work at Cold Spring Harbor Laboratory on Long Island, New York. And finally, when other people, the more genetic analysis became available, it was recognized that her findings were way ahead of the time. And she's one of the few women who has received a single unshared Nobel Prize in 1983. That's one of the equally prominent scientists such as Watson and Crick in the history of genetics. And these are the jumping genes, that were discovered. And you and I are here because of jumping genes, that the jumping genes played a part in human formation of human placenta. And there's something called the stroma and decidual cells that makes the placenta receptible to with the fetus, and it gets implanted in the uterus. And as you can see, this simple diagram shows how many of genes are retrotransposons. By retrotransposons, I mean that these are the ancient viral elements that have somehow made peace with our human genome and have inserted themselves and have lost their ability to cause disease and have become part of the human genome. And you can see that the retroviruses in humans, a large portion of them are, and the jumping genes come in, and there are the so-called retrotransposons come in a DNA transposon and a retrotransposon. So, and a simple way to explain that would be DNA ones are sort of cut and paste. I mean, you do that in computer or in a Word document, while the RNA ones or the retrotransposons are the one can be called as copy and paste. Okay. Or to put it in simple words that one can explain, is that so-called on-the-fly editing. Okay, and these were again, discovered in the work organism, the fruit fly. And geneticists have at least some good sense of humor that they can be labeled this. If somebody discovers a gene, they're allowed to name them. So the varieties of names that have been given to these fruit fly genes, which some of them would include Gypsy, Evel Knievel, Jordan. Okay, so all the genes that have flying abilities, so to speak. And these have played enough role in causing pruning of the nerve cells in early brain formation. Okay, because after a baby is born, the neurons go into hyperaction, and there are a huge number of neurons that form within the first 3 years. And as they are not utilized, they are pruned and kind of put in proper perspective. So from like 120 billion genes, neurons, they get down to about 85 billion. So some of the pruning that takes place is maybe due to retrotransposons. They do have a role in immune response as well. Okay, and the big role that takes place is in forming a protein called syncytin, SYNCITIN, and that came from retroviruses, and that has been used, in formation of placenta and preventing the immune compromise that takes place between the maternal gene and the fetal gene. Because as you know, fetus has both genes, father and mother, so to speak. And in spite of the concept of reproduction as being all such a lovey-dovey relationship, it is actually an evolutionary arms race that takes place between the fetal genes and the maternal genes. And we're just starting to read about, there's some other stuff that I've just read, which I don't know if I have that book here or not. I think, yeah, about evolution and Darwin. This is written by an Englishman, I think. So he talks about how some of the miscarriages that take place more during first pregnancy, or if there is early pregnancy that takes only after a few months, a few weeks of uninterrupted intercourse. And the reason might be because the genes of the fetus and the father may be rejected by the mother, because there is something called HLA, which if you've taken Dr. Spector's class, you may realize these are called human leukocyte antigens. And so these are not identified by the mother's immune system. So they are not accepted, and there is a lot more chance that the embryo would be rejected. So, and actually this blows my mind, so to speak, as to how this, okay, let me get this book, see if I can just find that. Okay, I think I have that somewhere here. So, okay. And it talks about how the success rate, oh yeah, I'm sorry, it fell on the floor, that's fine. Okay, yeah, this is the one that's just absolutely, this is well-written book type of thing. So I don't know if everybody can see that. Yeah, I think it should be able to see, but it's called Body by Darwin, How Evolution Shapes Our Health and Transforms Medicine. And he writes well because he starts out with a case history as to how one woman goes through 9 miscarriages, and that has simple cure about the rejection of the immune system that takes place, and the varieties of interesting cases. And then he goes on to explain as to how evolution has played role in our, our being type thing. So it's an interesting story as to how, and this surprised me that the success rate, of with uninterrupted intercourse is rather poor actually compared to cows and sheep. The humans' reproductive success rate is nothing to write home about. So again, we're digressing. So a lot of the viruses have made home for millions of years in human beings. So okay, all of us remember the songs by George Harrison, "I'm the Walrus," but perhaps he should have been singing "I'm the Virus," so to speak. So anyway, so the variety of segments that have taken place, of virus inheritance. And this is a nice example of a rather complicated slide as to how, the placenta, these are the retrovirus gene. There are the various segments that have been isolated, and how the protein forms, okay, in mammals, so how this goes on to form. This protein is incorporated into this protein that we talked about, this viral element forms into that. And this is a cat example. So this went on, about 60 million years ago, so that took place, and how the 2 cells fuse, and the placenta forms, and the maternal blood vessels come and incorporate. These are the so-called spiral arteries that takes place, that where the fetus literally sucks the nutrition from mother. And it's like an arms race. The mother wants to minimize that so that doesn't oversupply at the health. On the other hand, the fetus wants to get as much nutrition as possible. And there are some of the theories as to why the birth takes place when it does at 40 weeks, because there is an energy imbalance that takes place. For example, because there is only so much nutrition that mother's body can provide, and that may even be a reason for the so-called gestational diabetes that takes place, that the mother's body goes into hyperproduction for sugar, because the fetus to grow needs sugar as a nutrition type thing. So that may be one of the reasons for a lot of women getting diabetes during pregnancy, and it disappears after delivery. So this is this gives you an example as to how the virus and birth have been interacted. And there are various, if you may, at least those of you who have gone to medical school may remember some of these anatomy that we had to learn about trophoblast and varieties of protein that takes place. Okay. All right, let's see how far we are. So Okay, so there are a lot more technical details that will be in the notes type of thing, but I'm not going to belabor the point type thing. So I think let me go through a couple of more points and then we'll take a break. Okay, and so this is, again, this is a very scholarly article about how the whole microbiome is interconnected. Okay, like, these are the transposable element. Not all viruses are pathogenic. This is the gut microbiome that you may have read about quite a bit, because that has to do with your probiotics, which is a multi-billion-dollar business. At the same time, there are almost 100 times more viruses and the phages that are in your body that are there, because the bacteria have their own parasites, which are viruses, and some are living as parasites, some are commensals, okay, and they're mutually dependent. So these are some of the elements. These are the so-called transposable elements that are inside your gene. And if you recall from last time that the varieties of things, the so-called junk DNA that we used to call, these are the so-called transposable elements. These are the viral, and they're not useless by any means. They control the expression of the genes and silencing of the varieties of genes. Okay, and so these are the retroviruses that have made peace, which we talked about earlier. Okay, all right. So and so these are the inserted in two long interspersed elements and short. So these are they're associated with a variety of diseases. Let's see, I think these colors are rather faded, Okay, so these are the definitions of these long interspersed segments that are in your genomes. Okay, all right, the code for various enzymes. Okay, let's see if we can see. Oh yeah, the diseases, that's, so some of the diseases that they may be associated with are hemophilia, some of the colon cancers, autoimmune diseases. So they're not all the time always benign, so they can cause some plate. Now this blew my mind. I just read this morning, okay, because there are several science-related websites that I follow. This is about as hot off the press as you can see. This is 2022. It's just SARS-CoV protein spike activates the envelope gene. This is one of the small segments protein that we saw in the retroviruses that is encoded by human endogenous retroviruses. Okay, and why it's called W. Now, this is nothing to do with the big W that was in the movie. It's a mad, mad, mad word, if you remember from the 60s. But in biology, this is given an abbreviation for an amino acid called tryptophan. So this particular tryptophan RNA is used by this particular protein. So not all COVID protein had activations, but some of these had. So this is this. So the relationship in now blood cells. Now these are not red blood cells. These are type of white blood cells they showed. So this is the spike protein related article. So the active research is still going on as to how these retroviruses play a role even in the present illnesses. So this may have a role. I haven't had a time to look through the whole this thing, but because I haven't been able to find the original article yet, but I'll have some more information on it. So this is about as hot off the press as I can find for you. Okay, I think we're coming up to 2 o'clock, so we probably should stop for a few minutes before we dwell into something called epigenetics. Okay, and then I'll have more slides as we go on about diseases and genes as we go along. So I'm trying to incorporate both factors that we talked about. Okay, so I think, let's see if I have my time correctly. It's at 2 p.m. exactly, according to my desktop. So can meet in about 5 minutes. Okay, I'll get a sip of water and be back. I'll stop my screen share for the time being. Okay, folks, and let me get a glass of water and then I will sit around. Oh, I should pause my recording. Okay. Zoom does thing, sometimes they have a round button in the middle. Sometimes it's in those 3 dots you see at the bottom. Okay, so I think I've started the recording, so I didn't want to forget it. So we will talk about the science of epigenetics, or something called the Lamarck's revenge, Okay, and this is an interesting thing, that this is a classic example in genetics, that has to do with the cat lovers may recognize this is the calico cat. Okay, And, you get the two sets of chromosomes type of thing from, male is XX, XY, female XX. So female get the double amount of X chromosome. So the second X chromosome is turned off in the female. And so-called dosage compensation. So you don't get 4 copies. Okay. So this is, it's get to be, so this is something called genomic imprinting. So during the early stages of conception, this was discovered, and there was something called the Barr body. It's a phenomenon called genomic imprinting. So the gene is there, but the effect is so-called silenced. So that is what I meant by the expression of the gene that is turned off. The colorization of calico cats is a manifestation of something called X inactivation. The black and orange alleles means that the different forms of genes reside on the X chromosome. Okay, for any given patch, inactivation of X chromosome carries only one allele that results in the fur color of the other. So this is, so this is a classic example in, genetics about how the genes are turned on and off. Okay, and so this is the so-called Barr body, nothing to do with this part. And there are varieties of genes that are turned on and off between adult and newborn. So this is, so it's a lot more complicated than just this one X chromosome. So the names are immaterial, but say it's like in how pups, the varieties of genes are turned and off for social behavior, memory and cognition, and sleep, so in suckling. And the classic example that we said about, that as to how the mothers that provide affectionate upbringing is they lick the pups and they move the pups to the other cage type of thing. And then they are isolated the genes inside the pup. And those pups that were licked by the moms, they had the genes that were activated type of thing. So they were able to prove as to how the external influences, the so-called environment, influences the upbringing and the future behavior of the pups. So these were obviously, you can't do these experiments on humans, but but these are done on pigs, and it has been well shown that how, and so this is and now there are some examples of this that takes place, that there are some mutations that takes place in humans, and that causes, the similar abnormalities in epigenetics. And the classic examples of imprinting disorder are something that are called there is a one rare syndrome that occurs in 1 in 100,000 called Prader-Willi syndrome. And these are the kids who are horrendously, they become obese at a very young age, so much so that they have actually food hunger. They would try to, they overeat and become obese, that's called Prader-Willi syndrome and in genetics. And that is done with so-called imprinting disorders, means that the gene that controls their appetite and neurogenesis. So they have some retardation and some other these things. So, but fortunately, these are very rare diseases. Okay. Now I think I'm going to go quickly through some of these slides because some of these are very, very academic, and I think if you are interested in real serious neurosciences, then you can look through them. But these are, and I have already talked about the heart, the human activated genes, and we talked about the chimpanzees. So these are the varieties of functions that are activated in genes, and so human evolution is, and the various brain sizes that we talked about, how all the brains are made the same way, okay? And we talked about the various brain sizes in various humans, as to how the modern humans are different. Now, interestingly, this may shock you or disappoint you, that the brain sizes actually in the modern humans is slightly shrinking, type of thing. So the future may not be as rosy as we may want to be, Okay, and these are the varieties of genes that, again, they have relationship to memories, and again, the retroviruses have a lot to do with it. They are the shape of the HIVs, and they have to do with the memory formation that takes place. So this is the gag protein that you may recall that we talked about with the retroviruses. So again, as to how this particular gene interacts with this retrovirus protein and have much to do with the formation of memory. So how life is interconnected, that is the message that one wants to get across as to formation of memory, as to Okay, so these are the varieties, sequences that were analyzed, going again back to primitive organisms such as mouse and fly, and that they were found to be very similar to the viral protein called GAG. So again, this is the retroviruses have a long reach going back, okay, and so this that transfers the genetic material, type of thing. So it showed that decodes from an ancient copy of gene, which is called gypsy retrotransposons, if you recall the name that I gave you earlier. So this goes back to that type of fruit fly gene. So that has still playing a role in formation of memory in mammals. Okay. So we owe a lot to viruses. They are not all causing like a AIDS type of disease. So that's the message that I want to convey. I think so. And again, so they're like so they make from the independent copies. So they've made the piece inside your genome. And so we talked about the increase in brain size. So these are some of the redundancies. Okay. And again, so these are some of the copies of the genes that we remember when we talked about the human and the monkey chimp gene, the varieties of gene that we talked about, which are, the name again is not that important, but it has to do with the synapses formation as to how we start differentiating between the primate genes as for the human genes. Our brain is a lot more complex wiring that takes place because the growth continues, while in, say, it just looks like a stunted tree type of thing in the rodents, while ours is a lot more luxurious tree with more branches. So it's not the number of genes that counts, but what it does in the end, that's the big thing as to how this one particular gene has promotes the growth of synapse formation. And the classic term in neuroscience is cells that wire together, fire together. This was the famous statement that was done in the 1930s by the founding fathers of neuroscience. Okay, and at that time there was a huge controversy in the neuroscience field that was going on as to, are we dependent upon chemicals or electricity? Okay, so and turns out both of them are right, because there is obviously an electrical potential from your brain that is well recorded in EEG or brain waves. At the same time, there are chemicals such as dopamine and serotonin and acetylcholine. So there's also both theories were actually correct, But the whole so this was okay. Hebel and Wessel's famous statement: the cells that wire together and fire together. So they did a research in very primitive organisms and isolate the fundamental electrical properties of neuroscience. Okay, so this again shows as to how one gene can influence the growth of human brains. So this was the more anatomical microscopic evidence of the human memory formation and brain growth. Okay, and a lot of this thing is done with something called the gene duplication. So again, to repeat myself, that the nature uses what she has, which is the gene duplication. That is the big mode. And the second was the jumping genes. So just the evolution is an extremely slow process, the Darwinian evolution. But these two shortcuts is like moving into the fast lane that we talked about. So human-specific gene duplication has emerged as a modifier. And this is a fairly recent reference from the prestigious Nature magazine. But at the same time, if you, and some of the genes that have multiple copies and have abnormalities that lead to, if you have a lot more copies, sometimes it leads to autism spectrum disorder. And some of the theories that are being made for autism, that some of the genes and the neurons are not eliminated. And so their brains are, that's one of the reasons why they may be a lot more sensitive to noises and external stimuli type of thing, because the pruning that should have taken place in childhood has not taken place. And macrocephaly, so the brain is not actually physically bigger, but there's a lot more wiring than one needs to have. At the same time, if there is deletion, so that may lead to, and the faulty listings that may have a lot to do with schizophrenia. And obviously, it's not just all gene dependent. External influences or even your gut microbiome may have a lot to do with it. So it's a multifactorial thing. It's to and the book that I talked about, The Body by Darwin, okay, he gives a classic example of a man who is not a scientist, but his kid had severe problem with autistic behavior. Okay, and they were almost ready to send him to a special place where they couldn't look after him because he was interfering with the other children's lifestyle. And they decided, with some hesitation, to send the boy to a camp. And when he came back, from the camp, okay, for special children with autism children, they were sent, and the kid's behavior is entirely normal. He describes the case report, which is almost gut wrenching, as to say, okay, well, he went to McDonald's, had a nice conversation, and when the father looked at the boy's body, he was bitten by various bugs, chiggers or something like that. I forget the actual bug, and so the parasite changed the boy's immune response in such a way that his brain had quietened down. So how interaction took place between the autoimmunity and this may have a lot to do with, the dirty older brother hypothesis, the so-called hygiene hypothesis, that we may be a lot more clean than what we were meant to be. And that may have a lot to do with some of the autoimmune diseases that we've talked about place, And the same analogy applies to diseases such as Crohn's disease. Okay, and this may come as a shock to you that some people who had Crohn's disease, that initially they were treated with simple drugs such as sulfa and more complicated drugs, and recently the more biological agents that are used. But unfortunately, some patients have such severe disease that in spite of taking all this, the other expensive drugs, the one that you may see is Humira, that you may have seen advertisements on television, that's been around for 20, 30 years. Okay, and in spite of some of these new biological agents, they have severe Crohn's disease, which is pretty disabling, which is an autoimmune disease, causes diarrhea and almost bleeding and severe abdominal pain. And a lot of time the recourse is removing a large portion of your bowel. But some of the patients actually had done something called the whipworm therapy. That means intentionally ingesting drugs of parasite whipworm, which is ankylosing spondylitis for those who have studied parasites, which is a lot more common in developing world and causes anemia. Okay, so now obviously you can't just, so that they actually imported the whipworm eggs from outside US and dragged down the cocktail. And sure enough, these people's Crohn's disease actually settled down for several months. They had a rather dramatic response, similar to the response that you may have very well, I'm sure you've heard about, the C. difficile syndrome, which is treated with so-called poop therapy, okay, fecal implant, purified fecal implant, gut microbiome implanting type of thing. So a cocktail, and so that's, we talked about in the microbiome classes, I have talked about it, and so has Dr. Gerald Strelk, who did the history of medicine class. So, but that's, so there is a whipworm therapy has been, obviously, there are only very small case reports, but, Mr. Jeremy Taylor talks at length about it. And I'm surprised some of the work was done at University of Wisconsin in Madison. So there are varieties of these things that the gut microbiome has a role as to how interconnected we are with evolution, with the other species such as microbiome, parasite. And I gave you the example of toxoplasmosis earlier. Okay. Now, similarly, the genes turn things off and turn off. So this was one of the things as to how the embryonic limb bud formation talks, because the part of the genes that they are turned off and the activity is turned off, and that forms the limb formation. So this is the gene that has been well proven, something called the sonic hedgehog, as to how the web formation takes place. And there are certain chemicals that are secreted, they're called morphogens, and that controls the formation of various digits in human body. So these are the various expression cells, and by turning the genes on and off, scientists have been able to create animals with extra digits, or at the same time can turn on the formation of a thumb, so to speak. And this work was done in creating mutations, was done in the early 70s by a husband and wife group from Norway. Okay, names are, you may not be familiar with it, Christine Nuslin Fallhard and Eric Wickhouse. And so they were awarded Nobel Prize in 1995 for their discovery 25 years ago about genes that control the segmental development of anterior, posterior, the front and the back, and the side to side morphogenesis, means the formation of the body parts and the segmentation that takes place. And that was one of the genes that, the formation of the body that controls. And sometimes, similarly in snakes, some of these genes are turned off. So that's how snakes don't have digits and fingers. So this, again, these genes are conserved type of thing. So this is genes to fingers. So this is how the formation of the body parts is well orchestrated development that takes place. And we talked about the Hox genes that takes place. Again, I showed you this slide earlier. I should have silenced that one. And this shows about the gene duplication. So this is a nice video about, and the classic example of gene duplication is the trichromatic vision. That the red, green, and blue vision that takes place. So that, okay, let me see.
As we have seen when we talked about the emergence of multicellular animals, a very important mechanism of genome evolution is the expansion of gene families, groups of genes rather similar to each other. Gene families are quite frequent in today's genomes. They are the result of duplications of entire genes, and what is really interesting is that each member of the family can specialize in performing a particular function or working in a different cell type. Generally speaking, when a whole gene is duplicated, usually together with its regulatory elements upstream, there are several possible outcomes. The first and simplest, especially in the absence of clear evolutionary advantage for the gene and its protein, is that the new copy, or the original one, is inactivated due to some mutation, so it becomes an inactive pseudogene and nothing happens. Another possibility is that the new copy retains the original function, so there is an increase in gene dosage and in the amount of protein synthesized. This has happened, for instance, recently in human evolution in the number of copies of the gene that encodes amylase, an enzyme that we need in order to be able to use starch as a source of energy. If the evolutionary pressure to increase that protein is strong enough, as in the case of amylase, then we will see how the number of copies of this gene increases in the genomes of that species. But finally, and this is the most interesting part, at least to me, if the original copy retains its function, then the new copy can be used to experiment and acquire some changes that will lead to a new function or will change somehow the way in which that gene works. This process is called neofunctionalization, when the new copy takes on a new functional role, or subfunctionalization, when the gene does pretty much the same thing, but there are changes in the regulatory elements that control its expression. So each copy will be active in some particular tissue type, for instance. A good example, a classical example of this process is afforded by the genes encoding the globins. Globins comprise a family of genes, a family of proteins, present in different versions in all kinds of animals. When we compare modern globins, we see a picture more or less like this one with a hypothetical ancestral globin about 800 million years ago, that is before the arrival of animals, which duplicated to give modern neuroglobin on the one hand and all the other globins on the other. Then about 550 million years ago, during the Cambrian explosion, there was another duplication which originated modern cytoglobin and myoglobin, and a later duplication about 450 million years ago that led to alpha and beta globins. These, the alpha and beta globins, are actually gene clusters because each of them underwent subsequent duplication events, so that we have several alpha globins and several beta globins. It is very important to understand that these copies in the same species, myoglobin, and alpha globin and beta globin, for instance, are not the result of speciation, but of duplication. So all these globin genes in one species are known as paralogs because they are the result of duplication and divergence, whereas the versions of one of these globins, let's say alpha-1 globin, for instance, in different species, imagine mouse and human, are known as orthologs. These are the result of speciation. So gene duplication has been one of the main forces at work during genome evolution, creating gene families that can perform a wide variety of molecular tricks, enabling organisms to become more and more complex and sophisticated. And there is a final type of duplication that seems almost impossible or extremely unlikely, but in fact has happened several times over the last 500 million years, and that is the duplication of entire genomes.
Okay, so you probably knew more than what you wanted to. And this was the classic example of hemoglobin that, if you recall from last time we talked about, this was used as molecular clock by the famous Linus Pauling, and that started the clock of timing, trying genetic medicine with the fossils and paleontology. And so he used hemoglobin molecule that was even before Watson and Crick. That was the Linus Pauling. So I'm just repeating myself because it's an important point. Okay, and so this was, a lot of the work was done by a Japanese scientist also named Susumu Ono. So this was, a lot of the genes have been duplicated. And again, so nature is frugal in that sense. It doesn't discard, but just reuses the same thing again and again. So it's like a used car. Okay.
Question?
And now we'll talk. I'm sorry, go ahead, please.
So to make sure I understand, if the number of chromosomes in a species is defined, the number of genes is variable? And if there's no fixed number of genes or genome?
No, it's the proteins, because the genes are like, there are 20,000 genes in human beings, type of thing. So that's just, but they work in collaboration with each other, so to speak. So they're working, like, for example, all your cells would have the same genes, but the liver cell is smart enough to produce just the enzymes to digest your food. At the same time, your brain cell is smart enough to, just to code your memory. So, the numbers are the same. Basically, it's like the same orchestra that's given in different areas, but one plays Beethoven, another plays jazz. Okay, so it does a different thing because the permutation of combinations that they can do in terms of amino acids and proteins. It's like you got a lot more proteins and a lot more cell varieties, so they are turned on and off type of thing. So that's that's how it works type of thing. So because if you look at the cell, they don't have same chromosome, the same number of genes, but how they do, what the function. So that's what's meant is activation, suppression, working in collaboration. So that's how the whole machinery works. That's why it has been disappointing that even though we have the human genome code isolated, we still don't have some radical dramatic therapies. Okay, and even the so-called gene therapy, with all the enthusiasm that we have, it hasn't really, we've been, it's like 30 years or 20, 30 years since, with the Human Genome Project, was during the Clinton era, we thought, okay, we have found the secret of nature, but we haven't made, we have been only a few people who have been treated with sickle cell and few vector therapies that have been done so for single gene therapy, but at least we haven't been able to find a cure for diabetes or heart disease, type thing, because it's a lot more, and that's why there is a whole epigenome project that is being proposed as to determine how the little minor changes of small chemicals that are being taken place. And so we're just beginning to learn the interaction between our gut microbiome, the epigenome, and the various proteins that takes place, and the RNA. So it turned out to be finally, so it got to be a lot more complicated than isolating the 20,000 genes. Okay, hope that puts some, it's a long-worded answer, but it tells you that it's, and even and I have some slides about for schizophrenia and other stuff as to how we've been trying to tease out the answer in genetics, and with the science of computing, people have gone further and further. So, okay, let's take bigger and bigger data samples, so see if we can find some common thread. Okay, and that is what is known as the so-called polygenic scores. And and that's why I think I'll have probably next time I'll have some more slides about so-called evolutionary medicine that has been disappointing for, say, long chronic diseases such as schizophrenia or diabetes. It is being used somewhat for heart disease now, because the treatment is being guided, by whether it will be a mainstream or not, to do polygenic scores. But that remains to be seen, type of thing, as to how useful it will be in a practical sense.
Thank you.
Because okay. All right. Now, the skin pigmentation has been well isolated. There are varieties of melanin is the pigment that we talked about, the so-called two types, eumelanin and pheomelanin, that the red and yellow form, and the various shades that take place. And you're seeing human beings come in all sorts of colors. And there is a well-known, respected scientist named Nina Jablonski, who has spent her whole life doing research on the skin pigmentation and the complicated dance that takes place between skin pigmentation, the various genes that the melanocortisone genes that take place, along with the need for vitamin D and folic acid. Okay, because obviously, you need the ultraviolet light, UVB, okay, for formation of and proportional to light penetration. And at the same time, the ultraviolet light, destroys the folic acid. Okay, so the further away you are from equator, you need for vitamin D increases. So that's why you become lighter. And I don't think I put that slide in, because that's the color of skin pigmentation varies dramatically, type of thing, the further away you're from equator. Okay, and then again, so there's a little difference that if you go further out into the Inuit, they're more brown skinned. Okay, and you have to scratch your head as to see why does that happens. Okay, and because their diet is a lot more rich with fish, so they are getting natural vitamin D. So they did not have need for developing lighter skin. So really near the North Pole, so there was a totally different need. So a lot of things has to do with that diet, that the diets of pre-farming people was richer in meats and fish, which provided sufficient vitamin D. So the skin color was likely light brown as opposed to pink. But with the spread of agriculture, vitamin D became less from the diets of Northern Europeans. So reducing the melanin level, they could keep the nutrition balanced. So it's a trade-off between that took place. And interesting thing that the folic acid is important. So, and that's a lot more important for formation during the early times of pregnancy, so that some of the women who are strictly vegan and were not getting enough folic acid during their time of pregnancy, they were showing something called the neural defect called spina bifida. I mean, the spinal cord does not join, and it causes a serious birth defect in their babies. Okay, so they require, so that's one of the requirements. So that's why the pregnancy vitamins require the folic acid supplements. So this is so varieties of, so this is a very complicated, relationship that takes between vitamin D and folic acid. And again, so, and this has a lot to do with the formation of osteoporosis and earlier, and again, nature has a trade-off that during the childbearing age, the folic acid is a lot more important, while and again, at older age, if you get osteoporosis, nature doesn't worry too much about it, type of thing. So again, that's nature has her own way of making sure that the reproductive years are well cared for, rather than what happens at a later age. Okay, and again, so now I had some. These are some of them are out of sequence, so to speak. But I think these are some of the evolutionary things that take place as to how the jaw formation and teeth, because the teeth evolve. So these structured formed later on. So that's why the teeth are, malformation. A lot of us have malformed teeth. And these are some of the genetic evidence for, so I think this gets into a lot more details than see if you can read. And I mentioned a little bit about the this particular gene that is a pseudo gene. That's why we look like the baby chimps type of thing that we talked about, that we cause the narrower gene, and this gene has become so-called pseudo gene. Okay, means it is there, but it doesn't do its job. So that's why we have the smaller gene, the smaller teeth, and this third molar becomes less prominent. And this accounts for the the type nice jaw that we have instead of the prominent jaws that even the Neanderthals had. Okay, and so these are the inhibitory effect on jaw formation that has taken place. And so even though the genes are there, they do less of the job. So these are some of the gene formation that has been formed, I think. So based on how it is expressed. And again, this was the bigger thing about the eye that has always been the people were after Darwin that did not make sense as to how the eye formation is evolved, type of thing. And the eye formation is again dependent upon, let's jump over to, this is a gene called PAX6 gene, and the varieties of eye formation gene formation have taken place as to how it is morphed, as to how the human eye is different as opposed to the zebrafish and the fruit fly eyes. Because originally the eyes, the evolution took place from the fish onward, so it was designed as for better vision in the sea, and the color vision was at the depths of the ocean, wasn't much useful. So that evolved much later on. And this has been, the creationists have always had the argument as to how the eye is so complex that it would have required a creator type thing. So the varieties of these things that the eye formation has evolved in various different times as opposed to the octopus eye versus the human eye. Okay, and I think I did a class on the history of senses, and which we talked a lot more about the various types of eyes that are evolved. And so their eyes in humans are kind of extension of your brain, because your optic nerve is actually a part of the brain tissue that is extended. And so the images form backwards and upwards type of thing. And the aqueous humor, the light travels and it bends down, it focuses on the lens. The water in the eye keeps the eye puffed up, okay, type thing. So there are, and the various animals like some of the tadpoles and others have a layer called tectum lucidum, called a third eyelid type of thing. So, and so the eye formation is a lot more complex and has gone through varieties of nature has given different this thing. These are some of the fun facts about the eyes. They first developed in animals about half a billion years ago. First blue-eyed person, which I've said to have lived only, so it's relatively recent. All the babies are colorblind at birth, and it's a more common in male, red green color blindness. Okay, that is well known. And there is this is the classic example of the sex-linked recessive inheritance. As you may remember from classic genetics, there are varieties of gene inheritance that takes place, which is the autosomal dominant, that's the classic example being Huntington's disease, okay, autosomal recessive genes, okay, which you have to inherit from both parents. Now the third inheritance is the so-called sex-linked inheritance, that the females are carriers, and and if you need two copies for the disease to get manifest. So that's why, since males have only one X chromosome, that's why it's much more apparent in men, the red-green color blindness. Normal color vision, the chart is very apparent and color-coded, and the females are carriers, okay? And so that's, the daughters will be carriers, but the male child will have and it's very common in the classic example is the red-green color blindness. Sex-linked recessive, it's common in as many as 8% of male. Okay. And in some of the countries, color blindness would disqualify you from some jobs, okay, such as like pilots and train operators and so on and so forth. I don't think US has, and this was an interesting example of how the so-called founder effects takes place. Remember, I gave you the example of the internal brain vessel malformation. And this is the classic example that some of you are quite familiar with the famous Oliver Sacks. Okay, and in the movie that Robin Williams made about Oliver Sacks' discovery of the therapy for Parkinson-like disease, and then several books that he has written, type of thing. So he's a very erudite neuroscientist. Okay, and this is one of the books about the stories that he's written, The Island of Colorblind. Okay, so and that set up a medical clinic, in a small island in South America, and this was a documentary that was made as this with colorblind people, and the effect was due to, one founder effect that took place, and prevalent on this particular island. Okay, how the gene was inherited. Okay, and there's a documentary, I think, but it's just of academic importance as to how genes can come back, and so all the color blindness. It's a classic genetic story. Okay, why was it prevalent? Okay, this tells you the story of this slide. It's pretty self-apparent. Was one of the 20 survivors to have been a carrier for achromatopsia. Okay, and can trace their ancestry back to this particular person. Okay, inbreeding was necessary. That's one of the reasons why you don't marry your first cousin. Okay, so it just gradually kept increasing due to bottleneck effect and genetic drift. These are the classical genetic concepts. A form of genetic drift, the population is reduced, and the fluctuations in pairs becomes prevalent in an isolated population. Okay. All right, and I think we still have. Oh yeah, we still have a few minutes. Okay, I'll take a breather if anybody has any questions, comments. Excuse me. So. Okay, and these are the varieties that we already talked about, the evolution, but I think this takes us away from the classic example of the vertebrates and other things that we talked about. Okay. Now, this is interesting, and when we get down to the human body again, this is how the eating and evolution took place, like how the epiglottis, like how the larynx descended, in the human evolution, as to how in the babies, trachea is relatively quite small. It's only about 0.2 centimeters. So it's very small. That's why, and then again, even at 3 years, it's 0.5 centimeters, so it's again, by the time they're 12 and 13-year-olds, it's about 1.3 centimeters. So that's why the kids choke, especially when they start to eat with hot dogs, okay, or nuts, or with balloons, And finally, at age 20, the trachea becomes the size of 1 point. And initially, it's a very soft cartilage, and initially, so that's one of the reasons that the gatekeeper is an area called epiglottis. Okay, and so this is and this again has to do a lot with the evolution, because the amphibian evolution, breathing through the nose, mouth was under the water, and so the larynx developed type of a sphincter from something called the gill arch. Okay, and that was again prominent in fish. Okay, the various gill arches that developed, and the cartilage. So this has a lot to do with the evolutionary tree that we have inherited, okay, and again, so this is the example, and then the vocal cords that are at the top, they vibrate, and that gives us the varieties of speech, and you can show, shows the position of the vocal cords, okay. And this again, which shows the example of the infant versus adult evaluation of the things. Let me just see if I can. Okay, the speech, how the soft palate and epiglottis, and it lowers down, and the larynx slowly migrates lower down. Okay, and so the anatomy takes time to evolve, as to the horizontal versus vertical formation of the slip. So the high epiglottis, in fact, sometimes so that some of the newborn, the parents would look at the mouth of the babies, they would say, oh, what is this unusual structure? So the epiglottis slowly migrates downwards with the with growth that takes place. And that's one of the reasons why the speech takes time to develop, I think so. And again, a lot of this thing is controlled with varieties of genes. There is a gene called FOXP2, and that is conserved, amongst varieties of species. Let's see if we can get down to the, okay. See the gibbon gene versus various apes, okay, and how the gene formation has taken place as to of the so-called FOXP2 gene. And the classic example was a British family of Pakistani ancestry that had married the cousins and had the mutation in the FOXP2 gene, okay, and that family had that all of the children had difficulty with formation of the speech. They could not pronounce words such as blue versus they were pronouncing as boo. They could not pronounce the word spoon as boon. Okay, they could not distinguish words. Okay, so the mutation in this particular gene due to inbreeding, or the recessive gene became prominent, and there was a classic example of they were not identified, but they were called as KE family. And so it's a single gene caused such a huge abnormalities. Okay. And there is also a simple bone called, there are hyoid bone that is formed in humans, which is not connected with any other bones. And the larynx actually drops, and this gives us rise to the speech. And these are some of the examples as to the anatomical pictures that took place, how the evolution has taken place, how the larynx, and even at the risk of, choking, the speech was a lot more important for evolution, that it was a trade-off that took place. Okay, so how the different evolutionary pressures led to the origin of speech. And this shows to some of the goof-ups that Mother Nature or evolution has, that there is a nerve that is called recurrent laryngeal nerve that forms higher up and goes all the way down into your thorax. Okay. It forms from, it's part of the cranial nerves right at the top. Okay. Okay. It's one of the branches of the ninth cranial nerve. Okay. And that takes place, and it goes all the way down into your chest, winds around the aorta, and supplies the larynx. And that shows the common origin as to how it formed from the arches that took place. That's how the nerve takes this goofy place, And it's a classic example as to how, and it just makes the surgeon's life more difficult when they're operating on the aorta. So the left laryngeal nerve, so it shows as to how nature uses what she had earlier. Okay, and this is a classic example of the evolutionary trade-off that takes place. And similarly, in giraffe, you can see how long the recurrent laryngeal nerve is. It's almost, it's like several meters long, basically. Okay, so there are no shortcuts. So the vagus nerve just winds around the aorta. Okay, well, in fish, obviously, it's a small thing. So, and so these are some of the examples of that evolutionary trade-off. And again, the classic example of evolutionary, we have seen about the price that we pay for walking on 2 feet. And we not only talked about the backache that is common in 80% of the men and women, okay, but the same way evolution in osteoarthritis, so this is primitive new accommodative from walking to forced rest. So, the way it is transmitted through our hips and down to the ankle, and the accommodation. So when you look at the architecture of the, I think I may have eliminated some of the slides for redundancy and for time restraint, as to how the foot is kind of jerry-rigged, as to how the weight bearing is totally different. And obviously no successful engineer would design, because when you look at the South African runner who was running on the blades, he could outrun, and there was a controversy during the last Olympic. So that's the classical example. And this is probably the last slide before we stop, as to what is a vestigial versus atavistic organ. So many organs that have no benefits, that they are now called vestigious organs, okay. So, and so these are, still have tailbones, okay. many plants that so sometimes a mutation causes express itself that's got atavistic. So some humans are born with tails. Okay, and that's a very rare abnormality. So this is the difference in evolution that takes place, So okay, vestigial, yep. Yeah, so these are some of the ear muscles, this is that slide that we talked about, the science of evolution, that if you're interested in time, okay, ear muscles, some humans can actually move their ear, wiggle their ears, wisdom teeth, okay. Some of the male nipples, obviously the James Bond fans would remember, the movie, Mr. Scaramanga had 3 nipples, okay. So they're the kind of vestigial organs, okay. And spleen, okay, varieties of segment muscle, appendix, obviously the classic example, okay, it may have some from the coccyx tailbone, okay. So these are the varieties of vestigial signatures of evolution that are still left in our body. Okay, so I think we are pretty much in time. We are coming up to 3 o'clock according to my laptop, okay, and we should stop perhaps here. I'll stick around for a few minutes or as long as needed. And we'll see you folks next week. And I'll throw in more of the disease listing slides that I have. And obviously I'll send the slides along with the recording. I'll stop the recording now.